Hello everyone, welcome to the fourth module of the paper Cell Biology. In this module, we will study about the ultrastructure of the endoplasmic reticulum and Golgi bodies, which are some of the major cell organelles. This module would also give us a fairly good idea about the various functions of these organelles and the important roles they perform in the cell. Structure and function of endoplasmic reticulum. Eukaryotic cells are distinguished from prokaryotic cells by the presence of membrane enclosed organelles within their cytoplasm. These organelles provide discrete compartments in which specific cellular activities take place, and the resulting subdivision of the cytoplasm allows eukaryotic cells to function efficiently in spite of their large size. Ultrastructure of endoplasmic reticulum. The endoplasmic reticulum is a network of membrane enclosed tubules and sacs that extend from the nuclear membrane throughout the cytoplasm. The entire endoplasmic reticulum is enclosed by a continuous membrane and is the largest organelle of most of the eukaryotic cells. Its membrane may account for about half of all cell membranes, and the space enclosed by the endoplasmic reticulum, also called the lumen or cisternal space, may represent about 10% of the total cell volume. There are three contiguous membrane domains within the endoplasmic reticulum that perform different functions within the cell. The rough endoplasmic reticulum, which is covered by ribosomes on its outer surface, and the translational endoplasmic reticulum, where vesicles exit to the Golgi apparatus, both function in protein processing. The smooth endoplasmic reticulum is not associated with ribosomes and is involved in lipid rather than protein metabolism. Different roles of endoplasmic reticulum. The role of endoplasmic reticulum in protein processing and sorting was first demonstrated by George Pelade and his colleagues in the 1960s. These investigators studied the fate of newly synthesized proteins in specialized cells of the pancreas that secrete digestive enzymes into the small intestine. These experiments defined a pathway taken by secreted proteins, the secretory pathway, that is, rough endoplasmic reticulum to Golgi to secretory vesicles to cell exterior. The entrance of proteins into the endoplasmic reticulum thus represents a major point for the traffic of proteins within eukaryotic cells. Proteins destined for secretion or incorporation into the endoplasmic reticulum, Golgi apparatus, lysosomes or plasma membrane are initially targeted to the endoplasmic reticulum. In mammalian cells, most proteins are transferred into the endoplasmic reticulum while they are being translated on membrane bound ribosomes. In contrast, proteins destined to remain in the cytosol or to be incorporated into the nucleus, mitochondria, chloroplasts or peroxisomes are synthesized on free ribosomes and released into the cytosol when their translation is complete. Targeting proteins to the endoplasmic reticulum. Proteins can be translocated into the endoplasmic reticulum either during the synthesis on membrane bound ribosomes, which is called co-translational translocation, or after the translation has been completed on free ribosomes in the cytosol, which is called post-translational translocation. In mammalian cells, most proteins enter the endoplasmic reticulum co-translationally, whereas both co-translational and post-translational pathways are used in yeast. The first step in the co-translational pathway is the association of ribosomes with the endoplasmic reticulum. Ribosomes are targeted for binding to the endoplasmic reticulum membrane by the amino acid sequence of the polypeptide chain being synthesized, rather than by intrinsic properties of the ribosome itself. Ribosomes engaged in the synthesis of proteins that are destined for secretion are then targeted to the endoplasmic reticulum by a signal sequence at the amino terminus of the growing polypeptide chain. These signal sequences are short stretches of hydrophobic amino acids that are usually cleaved from the polypeptide chain during its transfer into the endoplasmic reticulum lumen. The general role of signal sequences in targeting proteins to the appropriate locations within the cell was first elucidated by studies of the import of secretory proteins into the endoplasmic reticulum. These experiments used in vitro preparations of rough endoplasmic reticulum, which were isolated from the cell extracts by density gradient centrifugation. When cells are disrupted, the endoplasmic reticulum breaks up into small vesicles called microsomes. Because the vesicles derived from the rough endoplasmic reticulum are covered with ribosomes, they can be separated from similar vesicles derived from the smooth endoplasmic reticulum or from other membranes, example the plasma membrane. The mechanism by which secretory proteins are targeted to the endoplasmic reticulum during the translational is now well understood. The signal sequences span about 20 amino acids, including a stretch of hydrophobic residues, usually at the amino terminus of the polypeptide chain. As they emerge from the ribosome, signal sequences are recognized and bound by a signal recognition particle called SRP consisting of 6 polypeptides and a small cytoplasmic RNA called as 7-SL RNA. SRP binds the ribosome as well as the signal sequence, inhibiting further translation and targeting the entire complex, which includes the SRP, ribosome and the growing polypeptide chain to the rough endoplasmic reticulum by binding to the SRP receptor on the endoplasmic reticulum membrane. Binding to the receptor releases the SRP from both the ribosome and the signal sequence of the growing polypeptide chain. The ribosome then binds to a protein translocation complex in the endoplasmic reticulum membrane and the signal sequence is inserted into a membrane channel. In both yeast and mammalian cells, the translocation channels through the endoplasmic reticulum membrane are complexes of 3 transmembrane proteins called the Sec61 proteins. The yeast and mammalian Sec61 proteins are closely related to the plasma membrane proteins that translocate secreted polypeptides in bacteria, demonstrating a striking conservation of the protein secretion machinery in prokaryotic and eukaryotic cells. Transfer of the ribosome from the SRP to the Sec61 complex allows translation to resume, and the growing polypeptide chain is transferred directly into the Sec61 channel and across the endoplasmic reticulum membrane as translation proceeds. Thus, the process of protein synthesis directly drives the transfer of growing polypeptide chains through the Sec61 channel and into the endoplasmic reticulum. As translocation proceeds, the signal sequence is cleaved by signal peptidase and the polypeptide is released into the lumen of the endoplasmic reticulum.